Dr. Shoma, go ahead.
Hi, hello, sir.
Hello.
Sir, it was a very nice, very informative, and the way you delivered your talk was very new, and we really enjoyed it. Sir, we are now moving on to our interview session. Sir, this is the part which I call getting to know our scientists better.
Okay.
It's high time scientists are more recognized for their work to make our world better. Sir, could you kindly say a few words describing your journey so far as a plant scientist?
If you ask how was my journey, it was like a roller coaster ride. It had an equal mix of both success and failure. I would just like to trace my journey all along the way. After doing my PG in agricultural biotechnology with specialization in plant molecular biology, I joined Directorate of Rice Research. It's now called as ICAR-IIRR, or Indian Institute of Rice Research, in 1998, with a lot of hopes to contribute in the field of rice genetic engineering. It was the in thing at that point of time, and also in molecular biology. But sadly, I was advised by my mentors to work on marker-assisted selection and molecular breeding in rice.
Okay.
Initially, I was reluctant. There are some lessons in it, but later realized that working on marker-assisted selection can open up new vistas. So what I did is I tried to see where are the opportunities. So I developed active collaborations with Dr. Ramesh Sunti's research group at CSIR-CCMB, and also with other mentors in India like Dr. A.K. Singh, Dr. Sudhakar, and others at different places like Tamil Nadu Agriculture University, and also with Dr. Swapan Datta's group at IRRI. I started working on genetics and development of bacterial blight resistant rice, post-pathogen interaction related to rice and its pests and pathogens, seed genetic purity assessment, cytoplasmic male sterility, fertility restoration, and necrosis, wherein I work mainly on gene identification and characterization and their utilization through marker-assisted selection. In this process, I realized that molecular breeding and basic genetic or molecular studies can be as exciting as genetic engineering. And I started enjoying my journey. Maybe in the first 3 years, I reluctantly worked on molecular breeding, but one should start enjoying their own work. And along the way, I got significant successes like development of improved Samba Masuri, development of a DNA test for seed purity, discovery of novel biotic stress-resistant genes and all. And in this process, I also got opportunity to be trained in some of the premier laboratories across the world and try to learn cutting-edge science from doyens like Dr. Ramesh Sunti, Professor Swapan Datta, Professor Revu, Dr. Jenna, Dr. Adam Bogno, and others. And these foreign visits and this training in some of the premier laboratories changed my outlook and helped to minimize my prejudices, because coming from a small city like Pondicherry, we always have our own prejudices and then mental limitations. And working along with doyens whom I mentioned, it helped me to widen my perspective and then widen my vistas. I learned the art of networking and collaboration from my mentors. I also, in the due course, realized the potential of new developments in science like rice genomics, other omics sciences, and then how they can make a difference in rice improvement, wherein we will be able to improve several recalcitrant traits. And in this endeavor, I learned a lot of good and bad things, and there were a lot of failures also. But I was not bogged down by the failures, and I'm very thankful not only to my colleagues, students, and mentors, but also to my family. My wife is also a scientist. She's a principal scientist working in Division of Endocrinology at Central Drug Research Institute. It's a CSIR laboratory. And she partnered me in my master's study in Pantnagar, and she also did agricultural biotechnology, but she later diversified to the other field. I'm also thankful to my parents and also my sister and their family for supporting me and then being behind me in all these efforts. And I would say that one should look at the opportunities, not at problems. Each and every problem carries along with it new opportunities. And it has been very satisfactory. And then I'm proud and happy to say that coming from a family wherein most of the people were working in medical profession. My father worked as a technician in the medical college. My sister was a trained medical laboratory technologist. I'm happy to say that I diversified and then started working on plant sciences or plant biology. And I'm very happy to and proudly to claim that I'm also a member in plant research community. Thank
Sir,
you.
we are also glad that you came into the plant research community. So otherwise, how would we get so many findings and so many inventions? Sir, my next question is that what new scientific approaches will be central to plant research in the near future?
So my answer, I'll try to frame my answers related to from a molecular breeder's perspective or a geneticist perspective. So one of the major tools which I think approaches will be genomics or other omic tools for basic and applied studies. I believe this will be very handy in understanding and manipulation of some of the recalcitrant traits in rice. I already explained yield is one holy grail, abiotic stress tolerance, and then tolerance to some of the recalcitrant pests and diseases. Certainly, genomic tools can be helpful, not only to understand the basic biology of these problems, but also to give us some durable solutions to manage it. And another tool which I'm banking on is in the next 5 or 10 years, genome editing is going to enhance our skill sets related to breeding, and also this will enable us to precise manipulation of traits in a very safe and effective manner. And this has been very aptly demonstrated by Dr. Kutubuddin Molla in his presentation. And I think that this will be the second most important tool or approach which will revolutionize plant research in the future. And there are other skill sets or tool sets like precision phenotyping, precision breeding, including laboratory information management software or field information management, and then large-scale application of data sciences or big data to both genetics and then plant breeding. And of course, as told earlier by Dr. Bharadwaj, genomics-assisted breeding will be the in thing in the near future. And added to that, if we can accelerate the pace of breeding through various tools like transformative breeding or speed breeding in rice, certainly our future is going to be very bright. And then I already see a lot of small-time laboratories and then medium-time research groups in rice breeding. They have started using some of these modern tools in routine rice breeding, and then I'm very happy for it. Thank you.
Yes, sir. Okay. Sir, my next question is, would you consider GM crops as an answer to the global food security challenges?
Yes, I definitely feel that GM crops are one among the few solutions available to us. I would like to underline the word few solutions because the problems are plenty. As on date, we face the immediate challenge of climate change and then other associated challenges like dwindling water and land resources, and then also dwindling soil resources. And we need quick solutions or quick tools to manage it. So I certainly believe that GM crops are one among the few solutions available to us. And my opinion is a judicious and pragmatic deployment of all modern scientific tools, including genetic engineering and genome editing. This can certainly revolutionize crop breeding programs and certainly ensure food and nutritional security. But my only submission here is there are other tools. For example, if already genetic variation is available in crops like rice, certainly we should go for genomics assisted breeding. For those traits where manipulation is limited or not possible, for example, breeding for stem borer tolerance or sheep blight tolerance, or for improvement of certain traits like drought tolerance or other things, certainly we should deploy conventional breeding, genomics assisted breeding, along with transgenic crops or transgenic breeding or genetic engineering. I believe GM crops are one of the solutions for meeting the global food and nutrition security challenges.
Okay,
thank you.
Sir, my last question will be, what is your advice to today's youth who are considering a career as a plant scientist?
So this is a question which I faced when I finished my master's, and then I was desperately looking to join a scientific career. So I would say that whether you are limiting your education in science to either graduation, post-graduation or PhD or postdoc, career in science and technology related to plant sciences is certainly one of the several career options available to you. This is because there's good employment opportunity in both public and private sector, particularly in cities like Hyderabad, which can be called as biotech capital of India, or seed capital of India, or agricultural biotechnology capital of India. What I find is there is lack of sufficiently trained manpower. This is one of the major limitations with regard to getting employment in both public and private sector related to plant sciences. When I go for conducting interviews for several public sector and private sector organizations, some of the prospective candidates, what they lack is employable skills or life-saving skills related to plant sciences is something what they lack. And as teachers and also technocrats, what we need to try to do is maybe we should conduct some refresher courses for 3 months or 6 months, either online, offline, or in hybrid mode, and then try to expose to some of the new graduates or postgraduates some of the latest developments related to not only molecular breeding but also genetic engineering and then genomics, and then make them more employable with regard to both public and private sector. And I would also like some of my younger colleagues is to have a habit of writing a diary of daily targets and achievements. And most importantly, enjoy whatever you do in science and work with full dedication. Failures will always be momentarily, and failures will provide you an opportunity just to know where you have failed and then how you have failed, and then try to minimize them in due course. And an important point what I would like to highlight is I have been fairly or reasonably successful in all my endeavors because of the good collaborations or networks which I have developed or which I am fostering. So focus on building up a good cohesive team of people wherever you are working. You need to be a good team leader, but also to be a good team player, because at various time points, you need to be a team leader and also a good team player. And you need to be true to yourself to science. Integrity or character is everything. And this is where I would urge all of you, try to be true to yourself and then publish only when you are convinced with the data what you have at your hand. And lastly, last but not the least, do not be bogged down by rapid developments in science. Do not fear change and try to adapt yourself to it. Try to keep yourself abreast with the latest developments in your field by reading latest papers. For this purpose, the initiative of Bioingene, and also there are various Facebook groups like Pant and other online fora, wherein we can get crisp information about the latest developments. Keep yourself focused and then try to download and then read the latest papers related to your field. And last but not the least, spend sufficient time with your friends and family, and do not bring issues or problems related to your work to home. Try to enjoy and then try to leave all your worries, prospects and other things in your lab or in your field, and then come home to your family with fresh, clean mind. Thank you.
So that's great advice. Thank you, sir. We are very privileged to have you here. Thanks for sharing your time with us. I'm now giving it over to Shubho. Thank you, sir.